So I think it was a great session, really starting from microbiome, going into the gut-brain axis, and then some really clinical examples. I think there's much to be learned, but I think we also made a lot of progress. One question always comes up, and that's probably a question for me, for you, Markus, and myself. The question about what is a normal microbiome, what is dysbiosis, and I give you the question is that how can we, if there's so many environmental factors like diet, genetics that could alter microbiome, how can we define a core microbiome, number one? And then I said that across aging, the microbiome remains stable. You mentioned in your talk several times that there's dysbiosis. How can we reconcile those two seemingly contradictory views? You can go first.
Yeah, I mean, I think considering that you are the expert here, maybe I should revise my statement. I mean, also, I guess we have to be a bit careful with, I guess, the term dysbiosis. It's a bit exaggeratedly used, and maybe you have to, first like come to a consensus what it actually means and how it's, under which condition it's happening. So I think maybe it's a longer thought process about this.
Yeah. Well, I mean, my opinion or my thought would be that I think we need to understand that in the beginning we had a lot of untargeted analysis, right? We're comparing something to something, and every single time there's a shift, people call it dysbiosis. I think, Holger, you mentioned a few papers. They only found really like one or two bacterial taxa, but they actually concluded the dysbiosis. And I think that's where it's coming from, that we need to define probably minor shifts, more shifts, like with every other organ system, like many different things change glucose levels, right? But I think we can have experiences with time that different shifts mean ultimately some relevance. And I think the same is probably going to the microbiome. So it's a definition. So I think we can probably both agree that in aging, there are some changes, but they're rather minor when you compare to disease or antibiotic use, and those changes can still be important, but I think that's where we have to come out. They still could be within a huge reference population, right? So that's, I think, it's a definition stuff, and so we need to really think. But I think defining maybe shifts is already very useful. You have a severe shift, then obviously it means something else. If you have a smaller shifts, then you probably have to look more metabolic pathways, that it's a multimodal, approach.
But don't you think it's more like the cautious postulate, like you have shown for GI disease? When it's present, you have the clinical signs, it's gone, you find that it's gone, in this case, real dysbiosis, and I think that's what's missing for the other studies, no? That we found something, but then it stopped, so we don't know if it's, rather, if the abundance is changed, what happens then, and can you then reintroduce it again? So not only change it to normal, but also reintroduce the disease.
Yeah, that's a really good point, because actually we usually see snapshots of but we are not following the trajectory and whether this has some impact on host health. It's usually just a snapshot and association. And
Yeah, I think that's really probably very important, those long-term studies, and I think we're learning more, like in GI disease in the past, for example, we have very short-term studies, like 2 weeks, 1 month, 2 months. Now we discover we probably have to follow them up over several years to see the long-term impacts. Perfect. Markus, for you a few questions. Is it possible that the choice of nutrients not only affects the microbiome itself, but also the choice of food items creates a kind of loop with the gut-brain interaction? So bacteria wanting the right substrate. That's maybe a question for all of you.
Yeah, there's indeed, it's a great question. There's indeed some preclinical studies which showed that that basically, and there's also some provocative hypothesis about who is the master and who is the puppet here. But there are some preclinical studies which actually show that the gut microbiome that dictate the behavior of the host to get this certain nutrient, and there are some studies also suggesting that the vagal nerve plays a role here in communicating, in orchestrating the host and making this choice for that nutrient or for that food pattern.
Maybe Holger, Natasha, do you know we have some nutrients that maybe just change the microbiome, but actually the effect is more direct on the enteric nervous system?
So the immediate thing I think of, it's not really a nutrient, but ammonia. you know, from a neurologist's point of view, anything that produces ammonia can be problematic, and that can be highly influenced by the gut microbiome.
Perfect. Maybe also, Markus, since you're an expert in probiotics, prebiotics, can you speculate on the mechanism of action of probiotics to maintain brain function in older individuals?
Yeah, also a great question. There's a couple of possible mechanisms which were also investigated by this research group. One is goes in the direction what we discussed already multiple times today: immune modulation. We have inflammation can lead to cognitive decline, and this B. parvus strain has some anti-inflammatory properties. In addition, there's some preclinical investigations that this B. parvus strain produces some indoles, which could possibly also have an effect. And then there's also something on neurotransmitters, I guess. I'm not cannot completely remember what the immune modulation and metaboloids were like hypothesized in this regard.
Maybe a follow-up question for you, Holger, because I know you did a probiotic study in dogs with epilepsy, correct?
Yeah, but the results are not there yet. Yeah.
The results are not there yet, okay.
Yeah.
So, do you have any anecdotal evidence that probiotics could be helpful in epilepsy, or is it because it's such a...
No, not really.
Okay.
Yeah.
Let's say with the microbiome, has there any research looking at the CSF metabolome in dogs and seeing if short-chain fatty acids are associated with epilepsy?
Not that I'm aware of, but it would be a good study to do.
Mm-hmm.
Have a look. Yeah. I mean, I guess the next is the question: is it the local change, or does it actually really go into the brain? I can't tell you. I mean, and the problem with the CSF studies, to be a little bit facetious now, is CSF is just like the lymphatic drainage of the brain, right? So it doesn't mean that what is in the CSF that's obviously closer than, and you don't have the blood-brain barrier, but you ideally would have to look at the extracellular fluid, and that was actually done in this experiment with the mice, and that's why it was such a nice study.
Mm-hmm.
When they did give the ketogenic diet, and then they looked at the neurotransmitters, they actually looked in the tissue, and that's very rare. Yeah.
Right.
I guess you could do a spectroscopy study.
Mm-hmm. We know there's some studies out there that show that probiotics can decrease anxiety. Is there anything probiotics and cognitive dysfunction, or is it an overlapping feature?
Not that I know of. Okay. Now, I think that now that anxiety used not to be included as part of the canine cognitive dysfunction, but now it really is. I think we're starting to embrace that, and studies will appear.
Perfect. And in humans, is there some probiotics that have shown to improve cognitive function? Maybe it's a Markus? Or Natasha, sorry, can if you know?
Yeah, there are a few studies, and with different strains related to bifidobacterium and lactobacillus, and so what I pointed out is B. parva. There are about 4 clinical trials existing, so this is the most studied at this point. Mm-hmm.
There's several questions with MCT oils. First of all, what is the dose of MCT oils?
Yeah, I mean the best effect was with 6.5% of caloric intake. If you go higher, the palatability goes down, and you didn't see more effect. Yeah.
And to follow up, do you adjust the dose based on the total fat level of the diet?
I mean, you have to take the caloric intake. So, I mean, in our studies, we replaced actually the calories amount by fat. That's how we did it.
Are there any potential adverse effects associated with feeding MCT long term?
We haven't seen anything. I'm not sure if Purina has seen anything, but we haven't seen anything. I have not reported anything. The only thing what has been recently been published in mice is that they changed the osteoclast activity. So there was a bit of a question if you have a patient with osteoporosis, if you need to be careful. But there's also no clinical evidence in humans.
So your patients, how many patients do you have like long term already?
Oh, we have a lot.
For how long?
I mean,
minimum?
since we have started 2008, so obviously this is RVC data, but also now at the TU since the last 4.5 years or so.
One question came up about recognizing cognitive decline. How do you account for general anesthesia?
You mean what's the
As a
question?
confounding
how
factor.
do you
So there has been a fair amount in the human field to suggest anesthesia may impact.
Yeah,
cognitive
absolutely.
function.
So general anesthesia and cognition in people is quite complicated, and we're just starting to scratch that properly, the surface of that in dogs. So in people, you can see an immediate post-anesthesia effect that can be dramatic but reversible, but now more and more there's concern that there's a long-term effect, that you've shifted someone on their journey into dementia, that you've actually shifted it. So there's kind of two groups that look at post-anesthesia cognitive decline in the human world. So when we started talking with them about designing studies to look at this in dogs, we realized it was going to be quite complicated. At the moment, we don't have embedded in our data, for example, how often has a dog had general anesthesia. We have actually started, we have an MRI study going, and we do some cognitive tests immediately before, then at 24 hours after, and 48 hours after, because we're like, well, our starting point is to see how often we can document that cognitive decline. None of the dogs have shown any cognitive decline with that. So I would say that we are at that early phase, but there are certainly some groups. There's one in Australia who've been developing a questionnaire. There is certainly increased interest about that, and there's no doubt that some dogs do get impacted negatively. The whole thing in people is, what are the risk factors to predict that? That's what they're trying to figure out. And inflammation is a big part of that, so the microbiota probably does play a role in that as well. But right now, we're still discovering.
A follow-up and a different question for you, Natasha. Can you comment on things you have seen that affect cognitive function from a dietary standpoint? So is there any relationship, diet in a bad or in a good direction?
Yeah, at this time we can't, because we haven't controlled what diet dogs are on. And the number of dogs, so we've got about 50, 60 dogs now that we've been following for quite a long time. And we have another 60 in a trial, but a lot of these dogs will pass away before they develop cognitive decline. So the number of dogs that we are able to prospectively follow, knowing what their diet is still pretty low. So not enough for us to look at associations.
Holger, do you have any experiences, diet and cognition?
No, I mean, I couldn't, we didn't have to do a prospective
So that means
trial.
you don't ask for dietary history?
We do. Actually, at the TIHO, they all have to fill out a questionnaire about diet since I started to work with you. So we look even at, Stan is here, we look at the poops as well. So, that's standard now. So we do look after that, yeah. But I mean, who knows? And I mean, there's one study being published in PLOS ONE where they followed them for a year, and the impact was very minimal. But the challenge is it might be interesting with the epigenetic clock. I mean, I think it's probably too short, and the effect size is probably not as big in a year's time frame. No, so that's probably the problem. And then when you start later, then you probably have too much brain lost. So ideally, you would probably have a couple of years study, like Natasha is doing, to really see an impact.
Marcus, centenarians. What do you think causes the microbiome changes in centenarians? Could it be due to the age of the gut, or diet changes, or other factors? Or probably?
Yeah, I guess there's many factors potentially. I mean, yeah. We're not really, it's again just a snapshot study, like we don't really know what it causally affects. It could be like, lifestyle, it could be environmental factors, it could be maybe also a certain genetic makeup, which make them maybe more successful, in addition to specific dietary aspects. Yeah. It's hard to answer with a clear response. Yeah.
Just for me now, I mean, because just pushing it back, but I mean, we talked a lot about the microbiome, but obviously the intestines age as well,
Exactly.
and they have significant changes as well, which are probably more difficult to study because you don't take any samples of them. But is there any knowledge in dogs and cats?
Not directly, I think, but that raises a very good point, and I often hear the answer when we see, we have this dysbiosis in chronic GI disease that is not reversible or persistent, and then always people are surprised because I think there's this thought that the gut is such a epithelium rapidly changing epithelium, so people think that if the GI signs goes away, that usually the gut recovers, but I think when you look at histology and function and the whole metabolome and the microbiome, all those changes persist pretty much forever. That means the gut is not functioning anymore, and I think it's pretty clear because the brush border reduces it. So you're right, the aging process is probably normal, but then if you have some inflammatory conditions, then you probably have this chronic restructuring, TNF-alpha, interleukin-17, constant inflammation is going to lead to restructuring of the architecture of the gut, and with dysfunction, I think that is something we really probably don't never think about. So the gut is very resilient, there's probably a lot of compensation, but the gut changes can happen, and then you have also microbiome changes, which probably going to go hand in hand to some degree.
Yeah, this is actually a really great point to point out the aging of the host and the gut, and this is indeed also something in these FMT studies where to look, does also these changes persist induced by FMT, or are they actually flipping back because of the environment, because of the host does not allow a persistent effect. Yes.
And it kind of maybe in line is, what is the effect of diet on the microbiome? And I think that's the same thing. I think diets changes their substrate. It can change metabolites and things like that. So we see those changes, but again, when you think from a biological system, it doesn't make sense that the whole microbiome shifts, right? It's going to be a shift that we can influence. I've always compared to a garden. If you have a garden and you fertilize your tomato plants, you're doing something with the diet, you get more tomatoes, but it doesn't change necessarily your whole garden, if that makes sense. And I think we need to find what is a huge shift, what is a minor shift, and still, we can still in a normal microbiome, more or less, still can very much influence some pathways that can be beneficial, correct? So that's the way I would see that. One question for you, Markus, about the FMT in the fish model, in this fish aging model. Was the time at which the microbiome is altered via FMT, did it affect the lifespan?
Yeah, good question. So indeed it is possible that maybe at a later time point, if they would have taken the microbiota from an older host, maybe, that it maybe would not be able to show this increase in lifespan. That's possible. Basically, they just looked on this one time point. It's only a speculation here possible. There's no data yet to answer this question, but it could be possibly that the age of the donor had here an effect on the chance on increasing lifespan.
There was one question about MCT oils. Oh, actually, it's probably a question for all of us, and I think we know how we're going to answer that. But when it comes to management of epilepsy and/or cognitive decline, what is most important in diets? MCTs, antioxidants, or microbiome modulation?
Well, I don't know. What is the most? I think it's different targets. No, that's at the end of the day. One is anti-inflammatory. The other one is giving a different energy source. But I mean, MCTs also have some anti-inflammatory component. And I think to be fair, and I mean, Natasha say, I mean, yes, a lot of diets and compounds have been looked at laboratory aging model, but also there have been some clinical trials, also placebo-controlled, and I think from the diets there is some solid evidence, but we are really lacking for drugs. And what is often missing is that it's just one compound changed, and it's often a blend, and that makes it difficult to really say it's now this. No?
Yes, completely agree. What he said.
Yeah, I guess there are responders non-responders. Some are responding to a certain nutrient or like a certain intervention, some less. Yeah, it's really hard to say, but maybe a multifactorial intervention, such as which could address different aspects. What I pointed out, Mediterranean diet. So this is more a dietary regime. So maybe it's more successful, but yeah, it's hard to answer.
And I think, I mean, for at least epilepsy and cognitive dysfunction, I'm sure it's a very complex disease as well. The only thing we know when we talk about, for example, idiopathic epilepsy, we have ruled out all the causes we can identify, but we don't know. We treat them all the same way, but you know, it could be very much like that the responders to MCT diets are the ones where there has been more alteration in the glutamate area, or they have more problems with the glucose. So it's not one disease, it's a bucket diagnosis. Exactly. And I think that makes it so difficult, unless we would really know 100% this is now because of this transporter defect or this gene defect or whatever.
Yeah, and I think we are in the era of precision medicine, and every individual is different. They have different issues going on, and while you can find common themes, really trying to characterize and understand what those different themes can be and where they are at in each individual will allow you to tailor the best intervention.
There's one question about impact of dog size or breed on microbiome composition. Does it relate to the speed of aging being impacted by size? Good question. Aging, I don't know. I mean, there are some studies that show small differences between different breeds and kind of would make sense to some degree. We have a small breed or large breed depending on transit time, passage, and so on. And at this point, it's still unclear what the effect is. Any questions from the room? You can just push the button. We can hear you. Oh, she's coming.
I think either 3 or all 4 of you had a picture in your presentations about butyrate going from the gut to the brain to the microglia. And I'm trying to understand the evolutionary reason for why would you signal from these fatty acids from the gut to the microglia in the brain. I mean, I can think of some ideas that maybe you want to know about your nutrient status, but I really would like the experts' suggestions about that. Yeah.
Yeah, we had this discussion before in the break. So, I mean, I guess one important point, at least to my knowledge, microglia, they don't have receptors for butyrate, but that not really answer the question. But I just want to point this out. There are trafficking immune cells, monocytes, which have receptors for butyrate, and therefore there is this interaction between butyrate and microglia. I was speculating about the fact that microglia, they have so many roles in the brain. They are like macrophages. They're eating things. They have a role in synaptic plasticity. They secrete BDNF and all these things. And this requires possibly a lot of energy. And where then they're wondering, the microglia, where we get this energy from, and therefore maybe they have an effect on, so where can energy come from? There are many opportunities, but one is, for instance, through the microbiome, through the diet supply. And so maybe therefore there's the physiological sense evolved.
Yeah, and I guess the pathway from immune response, it's also to make sure you don't get poisoned, right? So obviously the first part of defense is at that immune barrier levels, like Natasha said, 70%, and then we know the blood-brain barrier is perhaps not as tight as we thought, and then you need to have a second line of defense, so perhaps that's another reason for it.
To make life more complicated, there's also short-chain fatty receptors on the kidney. So there's short-chain fatty receptors everywhere. So I think it must be, the guts, I don't know. We have maybe time for 1 or 2 more questions from the room, from the audience. Sure.
And I think, Olga, you also mentioned something about immune cells. And what's been interesting, when I was taught immunology, we always thought that the CNS, or the blood-brain barrier, was an immune privileged site. That is literally changing as we speak. So there's now trafficking of immune cells that's taking place. And people are now showing studies whereby, and I think you mentioned it, about 70 or 80% of the immune system is within the gut. What people are now showing is actually there's a lot of trafficking that's taking place. And in particular, what's really making immunologists struggle is the biology of monocytes. And the idea now that macrophages, not only are they, you can get from the blood and then they come sort of from monocytes, derived glial cells, etc. But also the actual development, there's now evidence that they traffic to different tissues. And also the idea that this blood-brain barrier, there are cells that are going across, T cells are going across, etc. So one of the interesting studies that's come out recently is with multiple sclerosis to suggest that it's in some patients, there's a lack of Treg cells. So it's really becoming interesting how the gut and the influence of the gut immune system and how those cells traffic.
Yeah, we've seen the same for drug-resistant epilepsy.
Yeah.
Perfect. I think we are perfect on time. So if there is no super urgent question or comments, I think we're going to close this session. Thank you, everybody. It was really very engaging and very enlightening.